Everybody agreed?
Yeah,
okay.
this one as well.
Okay. Okay. So before I put the next slide up, I want to say that the reason I didn't want to answer anybody's question is there isn't necessarily a right answer for the order of these.
Oh.
There's not a right answer for the order of these. There's probably a wrong order, but there's not necessarily a right order. If you're being intentional about what you're doing and why, and you have a good reason for it, then that is probably okay. The most important piece is that you are doing all of these steps with a good reason. This is a really common way that it gets used, that the learning cycle gets used. So this is certainly a way that works very, very well, and it's the way that I design a lot of my lessons. But you can be flexible. You don't have to follow this exactly. And this is on Google Classroom too, so you have access to it. So even though this is the way that I usually design my lessons, there might be a reason why you want to do application before you teach any content. Or maybe before you let your students explore, you have to introduce some definitions so that the exploration makes sense. Sometimes, if your teaching is going really, really well, what the students learn from application can lead to the next concept invention. So you can actually have small cycles within the big cycle. That's a more advanced strategy. The basic strategy is to just go around the circle. But know that as you get comfortable, you can move around a little bit more freely. Any questions about the order?
Application kare? Yeah, he thinks isn't it better to have the application at the end, because when we learn something, we reflect deep on that and then we apply that in the real life. So application is better to have that.
It's a good question. The general thought on ordering it this way is you build your students' curiosity in this invitation to learn so that they're excited to learn, or they've been sort of invited into the learning process and they're excited to be there. exploration is where they get a chance to build on that excitement and also to tie it into their pre-existing framework of knowledge. So if I teach you something and you have no context for understanding it, it won't make any sense. Sorry, I'm not
हाँ
giving
तो
you
तंदा दे चिरगे बना कुन रोता चाचियो ते तंदा पानम तुस रेस तंदा छोरन को सोवाते डिगा डिग दूस रवा मैं तंदा चाचियो अंडु सदे देरेस ता तांपोते कलाना चिग थोप ठिक तुजु छंखोरि लोबजुन चियाला yang गुया तिरवा ता कुन रंबो चियां गुया तिरेस तो ठिला या चिग खुचुला चिग जाया के टुना लो या ता चिग समलो नने चिग जंदु को लुस चिया चिग रवा तेरेस ता ती की जेला या लोठि तेला या ने छोपसि चिग चिग रवा ता कुन नेतुन चिग तन जेवी के छोसि चिग चिग रवा छोसि चिवा तेला तेने देते लोठि के ता खुरंग ngeru gi shisha pecho na te tigi namba te kanza de kap sunga gures.
I'm going to try to get to your question. So exploration is sort of where you build the context or the foundation to teach something a little bit more specific. And that happens in concept invention, where you maybe give some definitions or some new ideas that they weren't familiar with.
Tene tanda ge nga de ge choship se de ge kapsa kare se na cherangal homchin chi trip pi ge ta tonchin che ta pa chi trip pi ge ta mangshi dang ya deres ja chi sana mangshi dang ne de ge jela ne tanda de ta che se to se do ta che she ta pa chi ne khu ge chi nang tun ta ngu karim batin de she jala ne ta deres.
And then in this application phase, you take what you taught and you have them kind of grapple with it. It's sort of where you give the hard questions that they don't necessarily know the answer to, so that they have to do their own learning. So after you give a definition, then they have to apply whatever they learned to a new environment, and that's where the learning gets what we call sticky.
Ta tanda de kaps sambati kaps nga suge ta de kaps invention session ma to piserto session ma to ngone tane sambandi theater do ku ne sapa khutro theater do ra ta nisu khutro che nisu te choma khutro che no ge jela ya ta de pesho se di kare no sana thoptrike ta chi thoptrila ya chi chare chano go jela ne jowa chi pe de kal khapu na no chi tang sha pa ba na thoptrike te ju pesho tang de tong sugro This pesho tang de ta total land de ko yo wa odis tambala de tan de pesho se ate ani trisha ju nang ton ti pesho tang de ta to khapola land de la so pa ne tan ti chag yores.
And reflection might be used slightly different than it is in a monastic situation. Reflection is generally a little bit shorter, and you're sort of asking your students, what did you learn? How did you learn it? So, and what that just helps is it, when a student writes, this is what I learned, and they write it down, it helps them remember what they learned.
Ta tanda ge samshi sa chi or wa nga chu samshi sa te chi che ba na ko lan tan te che chu ge ane chi gonde ta sang lo mye gi thö ge samshi sa te ta chi china manda ins baris tanda de kaps samshi sa te kare ko re sa na thopthi la ya chi kare jae be kandas jae be sa te la ya ko lan te res ta ke te thopthi ke kare jae be kandas jae be sa te ko ge sentri ga te lenge de jae de ha ko song lamna ane te ko je la ya ko zenda te kaps janju te temba so ya la phengi res.
I think when I'm hearing some people say reflection in a monastic situation, it almost sounds to me a little bit more like application, because you're thinking about how does this apply to your life. You're reflecting on how it applies to your life. That's slightly different than how we're using it here.
Ta chechu nyamlang ki thone te shuchambo te ga samlu shit tang de tang de machina tele chi pechu kandas tang gore chi yad ke yuan jela sopa ge chi yuan jaya chi or wa te ge ne tang den ru dus ta pechu kan te chi ya pechu tang de chi ge samlu tang tang de gan ru dus ta tinda ne chi ge tra jibu tin sabu tinda maimba tan de kap su ge che shi tan de samshi sa te tis mando res.
So the key part of application is your students are trying to figure out, they're working with questions they don't have the answer to.
Ta tanda de ge pechu sa to ga pechu sa de kare se na ne lob thila ko so twa khapu chi tang de len de ta len khapu yo ge twa de la ya len sa chi gorwa o de ge de ne tanda de ge pechu sa de ge tpcha ke chemu chi chagres.
Of the activity that you just did, what was the application phase of what you just did?
Ta tanda che chu ge chegu chi de barwa 제고 드립샤주 되게 아니 배추기 차시대 관계를 두스. Yeah, to order them in.
To order them, yes. Yeah. So even in this lesson, I'm trying to model using this lesson.
That digging the theatre to go read, digging out the language to take on patient answers.
Okay. And we'll also try to model this in the biology lesson that we're going to switch to now, because I think it's time to move on.
That Chingwegi, Joshi Chingoto Chigore, digging all young as with the Tandage, the Tandamoto Chigoroa, the patient against.
I think this is fascinating, and when I learned about this, it changed how I teach. So if anybody wants to talk about this after lunch, or at lunch, I'm happy to talk about this. As we mentioned at the beginning, and some of you also acknowledged, this is very, very similar to the 5 E's. It's the same thing, but with a different emphasis.
Gama de dan dan ing identity do yores in bana ko gi chi pe tan an chi ga mixin zik tan chi go wa kan ge chi ga muk yo mete mandore ma to pe an do res.
Okay. So as we go through the lesson about biology, pay attention to where we are in the learning cycle, and how are we using that to maximize learning.
So in the single channel Joshikola and Deshajago, Deshajan Stabalaya, Karituna Chirochi Sena, and a Chirapachi Tripic of Sangaso, Tanda Shijung, Korjing and Ramba Coach, Hedu Tetuna Chirochi, Esra, Te Le Call Hedu Tetunachi, and it tea Pechotan de Ting as to Comte Mando Mandochiora, Te Pechotan de Tang as to Hobjon Chia Teta, Yasuchia Segorata, or Te Laya Candas Spencer, Te go some Tanochis.
Okay. So, let's talk about biology, finally. Okay, so our goals for today, we're going to learn about the different types of cells. And we're going to learn about the parts of a cell. What I'm calling a goal, you all called... Oh, it's on something back there. Students, what's that?
Desired result.
Desired results, yes.
Or goal, there's some goal as well, yeah.
So it might sound different, but it's the same thing. Okay, so we're going to start with my invitation to learn for you all. I want you to discuss at your table, what is a bacteria and how is it different from an animal or plant? So this is my invitation to learn, sort of the entryway into our lesson. So just talk in your group for 2-3 minutes, and then we'll share out. As I'm reflecting on it, I wish I just made it the same with the 5 E's, because I shouldn't have introduced 2 things. I should have just done the 5 E's instead of the different labels for it.
It's okay. It's good because they need to be introduced to different pedagogical methods.
Yeah.
They can use Some people prefer one over the other, but
Sure.
I think it's better to have many pedagogical methods.
Thank you. I didn't come looking for comfort, but thank you.
Oh, no. I just really like all this just coming together.
Yeah, I'm trying to do that. I'm trying to make it come together. Okay, let's bring it in. I'm curious to hear what you all talked about. So you can just talk into your microphone. What are some differences between bacteria and other organisms?
따뜻하게 승부요로 승부다 네 지금 현대 별라 개발가요레스 메시지하르지사
ngotso mendo khabaranga dijirasro
aha
ani tombote ani tenyame bache tinyi
no
Tinyi
necklace
mepa.
they find 5 differences. The first ones they don't have necklace,
ani chopong chigdin ba nande mai ba
cellular,
ani chopong ko kimo me ba
no cell membrane, cell wall.
gelatin ke so gelatin ke ani ta gelatin ba koro ranguni ke me to ba gelatin ni ke
It depends on other organisms to survive.
Not necessarily.
Yeah, tane?
Yeah.
ani njokpo kemper che dongi lila ba
It reproduces really fast.
It can. Yeah, it can. Yeah, it can. Yeah, sorry. So yes, it can reproduce really fast. Sometimes it depends on other organisms, and sometimes they can be on their own.
Do they have cell walls, cell membranes?
Yes, they do have cell membranes.
Okay.
Yeah, and some of them have cell walls. Yeah. We'll get to this, but cell membranes is one of the things that all cells need to have. If you don't have a boundary to the cell, it's hard to have a cell.
The tapunga chimutinga so kalakala motor chigis, tapon la ya susu jendale loss gag chimuch gugu. The meba imbana tapon sayati tenisha kabuchig was. Tinga so kale dishes. That's enda.
Simbula one time or something. Shamba the chipa duration other than a simbula one time.
Singular?
One time.
One time? No organelles.
No organelles. Yes, very good. What's that? What's that?
ngazu tsobal chashana ani tingi yomar yena tingi gisale ya ninda daw jiyores tingi yomar yena dinsele ninda tingi daw jiyores chire ani korala mandawa ngama flagella la jo ngama mandawa jiyores.
Yeah, it has a flagella for bacteria, it has flagella, and they don't have nucleus, but they have something in place of nucleus.
They have just DNA. It's in the cell.
DNA, yeah.
Yes, so some of them have flagella, not all of them, but yes, some of them do.
Go on there.
Okay, good. Sounds like you all already know a lot about bacteria cells.
The simbugi tapon kola machi shigi mabuji haku deshas.
I'm going to go on break.
Sama haku deshas person tange insingara.
So there are generally 2 cell types. We're going to talk about 2 cell types today.
Da nga su shirgi imbana tapon la ring mando ani yores wa ane ti kola deshagins.
Bacteria are what we call prokaryotes.
Da simburi chiora dila nga su nyingte segres.
And then the other type of cell that we'll talk about are eukaryotes.
Jenda tapon ri jenda telaya nyingden segres.
This word prokaryote basically just means no nucleus.
Da de koranga inchi tsige nangtong karese na tinyang me pati kugores.
And eukaryote means it does have a nucleus.
Da inchi tsu shenda tige tinyang dem pasigores.
Prokaryotes include not only bacteria, but this other group of really weird organisms called archaea that we're not going to dive into right now.
That's chưa tanda nga su nhìn trek na la ya simbu chibu mai ba tele nga su architecture sa chiwa tiang na chu yores.
But they are, just for your information, they're fascinating organisms that live at all of the extremes on our planet. Like the coldest cold, the hottest hot, the deepest deep. That's where you find archaea.
Tanda architecture sa te ko kala chune chigu yores stambala ya zamlang genge ta nam shi tang tina do siguro da khori tipe sipsala. Betit hachang changsala niye yamna hachang chasala niye yang ko chugi ba na ko ting sapsala niye odinda res ku chuni chisate.
And eukaryotes are found in plants, animals, fungi, and they can also be single-celled organisms.
Da shenda tanda nga chugi nyinde ti kagina na yore sena socha che zishin che ani hambu yorwa de che ne shenda ko ya tanda nga chu thapong chik dengi chingu yorwa ti na yores.
But before we get into really learning or delivering a lot of definitions and things like that about prokaryotes and eukaryotes, I want to do my exploration phase.
Da tanda nga chugi nyingden ta nyingte ge ko ji kasukure da nang ton ta cherper tinde she majong ola ya tanda ngi choship se ti ngo chigi ins.
And if you remember, exploration phase is where you give your students an opportunity to explore on their own.
Da tanda ki choshi se ni gare se stambalal hom tanda ti ga param tin ne ta ba imba na kare se na thopti la ya kuchu ngo ne chi twitter chi ta kuchu ne chi chogi chi ga chi ro o ti gi rambati res.
So what you are going to explore are some slides that I brought, and these slides have both prokaryote and eukaryotic cells on them.
On each of them?
Each slide has both prokaryotic cells and eukaryotic cells.
Okay. Da tanda ge dela ya gela ge she chidwa she de gan la ya ko thapon yores thapon she chig gan la ya ane ko ninden ta ninte ni cha yores thapon.
So you and your table, I only have enough for one at each table, so you and your table will look at these and you'll do some exploration on your own, and I'll tell you what I want you to do in just a second.
Da tanda de kala na che chu rukha re ngola ya thaton chi she ju ji de de en tanda de she dina gala yo pe ge thapon la ta res ane tande chala ya dosi re gala ya she dina do re ma she thokumi dus.
Just like you did with cheek cells and onion cells and the mouse brain cells, I want you to do scientific drawings of one example of bacteria and one example of the eukaryotic cells.
Da tanda de kaps che chu ge ngola ya thaton chi she oni ta de ta chi na yorwa tanda nga chu ge kasa chong pa pa ta jamba damba na lo ne kon ju ge ane thapong re tini shenda chi chi ge le pa tu sta yorwa tu sta wi kaps ane kari thomba de rmoyang tri yorwa ti na shin tanda ya na ro che tanda de kaps kari yore sena nyindeng ge thapong chi ta nyinde ge thapong chi yorwa. So
one drawing of eukaryotes, one drawing of prokaryotes. The bacteria, one thing you will learn in doing this is bacteria are really small. They're small enough that they are going to be hard to see even with your most powerful lens. Especially if your lens is scratched. So do your best, ask for help, and use your best microscope.
Ane takatsu tutuchi naroches ane shola ya meshio kange tepa mbana ane ketanchi singamundu seropa ketanoches ane chi ta kanyonchi cheroches.
When you look at the slide, there'll be a big dark thing in the middle. Those are eukaryotes, and then there's a very faint green spot that will be your prokaryotes.
다 단다게 셰디간도 시타바이나 데나라 지기티스 나푸여서 낚시체여서 전부 지거레스 다 땡아츠그 인den기 타폰테레스 니 데나라야 지 전체 장코로 지셀라 마셀라 지거레스 데다 디카스 인제 임바간실레 심부리도가 신친데레스.
If you can't find them, raise your hand, ask for help.
아니 게티 치셰올라 타치 니금도나 니 케탄로치스.
And then if you can't find them, you can draw this instead.
단도 니금도나 니 르문디트샤나리레스.
This is an example of your cheek swab that has both your cells and also bacteria cells that live in your mouth.
다단다 데 트나디코레스 데 카레스 스탐발라에 토야테 코네 토레 초랑간 담바가 나올로니 토엔칭가 수타치고 아테레스 데 강라 카리도세나 초랑가 담바가 나올로니 토엔비기 초랑가 타폰라 도스 템세 니 숨부가 타폰치고라 bacteria 도스.
So I would prefer you use the slide. If you can't get the slide to work, do this.
다 담보텔라야 단다 gelala 포기 셰치고와 데 강게 텔라 타테 데르모트로치스 데 게티 촌에 얘기면도나
Okay, so let's have folks go get microscopes. I think we have enough for 2 at each table. That doesn't really help because you only have 1 slide, but you can use your best combination of microscope lenses to get the best image if you have 2 scopes.
약수티 배치당데 스거로다 게데 거기 지식이 셰트 지우자구나 지우자다 때녀서 소착으로 이나야 지우자지에다 간다만다다 약수티 배치당데다 데타로치스.
Let's take 15 minutes for this, and then we will move on to the next part.
나하다 까마 중화 배치당데다 단다 제고데로치스네 되게 제랑아서
They are there. You can't see them.
전전 직선치기 yes.
Ready, set, go. So the bacteria,
먼저
as
나로치스다.
you're coaching them through it, it's really stringy and hard to see. Yes, yeah, and really like the really long and stringy. They're like colonies of long stringy things. Can I just give you the slide real quick? So this is going to be the eukaryotic cells, and these are going to be the prokaryotic cells. The prokaryotic cells are going to be long lines of stringy bacteria that are all lumped together.
Okay, which visible one is.
Oh gosh, sorry.
Visible one is.
Eukaryotes,
Eukaryote and.
prokaryotes.
Okay.
Yeah.
Okay.
Yeah, I was just going to go around and explain it because they're. I figure if I give the person that's like going to be coaching them just a little bit of, you can do that too. Just tell them that the prokaryote, which is like the little thin green spot, is thin, stringy, hair-looking stuff.
Okay.
That it'll look stringy and hairy under the microscope.
So how many are including per
1 per table.
1 per table.
Yeah, I would love to give more, but I only got enough for 1 per table. So here's a slide for you all. These are going to be your eukaryotes. Oh, sorry, I'm in the microscope, or the microphone. Not everybody needs to know this. So this is your eukaryotes, and these will be your prokaryotes, these things. When you look under the microscope, they'll look like really thin hair. So if you see something that looks like really thin hair, that's what you're looking at, and try to zoom in from there.
This one?
This one.
This one? This
Nope.
one, how we can look?
This one is easy to find. This one will be hard to find, and I can help.
Okay, okay.
Okay. Eukaryotes, prokaryotes. Kind of hard to see. If you hold it up, you can see like thin green stuff. Very, very, very fine. It will look like hair. It will look like very thin hair, kind of green. If you have a hard time finding this, it's okay. Raise your hand, I'll come help. Okay. These are these thin.
Oh, they both on one.
Both on one slide. Yeah. this is going to look like thin, wavy, very small hair. It will be kind of hard to see, but if you see things that look like little hair, go ahead and try to focus on that and then zoom in. It will be challenging, but that's one thing to know about bacteria is they're very, very, very small. That's part of what this is about. Here you go.
Small one is the prokaryotic.
Yes. Yes.
This
This is the same one. Same thing. Same slide. I have an extra. You should take the extra.
okay, that's good.
You are the only table that gets two. You can just turn your, you can leave your microscopes out, but just put the slides somewhere where they won't get broken and the microscope somewhere where they're out of the way. So, what did you notice? What is the main difference between prokaryotes and eukaryotes?
The shape?
Shape?
Yeah, the one is like a thread-like.
Yes. Yeah.
And yeah. Also, the size, one's bigger, one is small, and
Yes.
one is also like so many dots, like so many cells
Yes.
in one place.
Yes. Awesome. So you're noticing that the size is different. You're noticing that the complexity is different, and you're noticing that the shape is different. Just to make clear, those threads that you were seeing are many bacteria kind of linking arms like this. So they are independent single-celled organisms that lock together. Yeah, like beads. Yeah, yeah, like this. So many individual cells strung together. Thank you. Okay, so that was our exploration phase. You got a chance to explore, look, and sort of get a sense of what the difference between these different types of cell was on your own. Now we're going to move into concept invention. This is the same as explain in the 5 E's. And this is where you start to teach specific ideas. So, let's go over the difference. First, we'll go over the difference between prokaryotic cells and eukaryotic cells in a little bit more detail. We are to talk about. So, first, let's talk about the ways that they are similar, and then we'll talk about the ways that they are different. So, both prokaryotic and eukaryotic cells have genetic material, DNA. They both have cytoplasm. Cytoplasm is the media inside the cell that everything else moves within.
다 특히 세대 갈라 타풍가 난게다 미디어스 타풍가 난게 접차데 제 되게 나라야네 타풍가 나라요 베젠다 참마티 줄리던 거 줬던 거 요레스.
They all have a membrane,
참말라 타풍치 뭐 요레스.
something that defines their outer boundary.
되게 갈라 타풍 코랑 그 실록가 산삼지 요로 참티 그 버지기 요레스.
And they all have ribosomes.
참말라 라이보타지 요레스.
Ribosomes make proteins.
라이보타지 클릭 가레스 스탐발라야 디지즈 튼제드레스.
It's not the official definition of life, but you could make an argument that life is just the ability to make proteins that sustain themselves.
단다게다 세속세나 세속세딩 속에 되게 딱고게 가서 용체게 대세다 마다 임바이나야 세속세데 가레스 스나마 수수수수군에 디지제튼지 뜻받지라야 데나야 디그레스.
It's theoretical and not super useful, so it's just fun to think about it that way.
그 남자가 되네 임바이나 데나로 진데세 자는 디고레스.
But the reason I say that is all cells make proteins, and ribosomes are what make proteins. That's the important part.
단다 데나네 지게 치시드가레나 타폰 사마게 디지티 조튼지기 요레스. 되게 나네 타폰가 나네 딱 그런 거 접차 네 라이보타지 요로와 되게네 디지제튼지기 요레스.
So genetic material in purple. So in this picture, you can see the genetic material for the bacteria, and the genetic material for the eukaryotic cell is in the nucleus here.
당보된 가서 토모도 옆에 당보된 데로와 르지로와 르지데 임바나 단단되게 선탕나는 다토탄 샤주도 아데레스.
The cytoplasm is just all the stuff, the media inside the cell, this liquid media.
다 타시티카레스는 나라 옆에 타풍 나라 옆에게네 셔즈우지고로 아데레스.
The cell membrane is this outer layer, and we'll do an activity that helps us understand the cell membrane later.
단다되게 타풍 그 지모요라 지모데 실로라 옆에데레스. 데 콜랑 아수게 제구치 듭기인스.
And ribosomes are these really small little dots that you can see throughout the cell. And they're very small organelles, and we'll go into organelles later, that make proteins.
단다게 라이보타즈세데 타풍 나라 옆에게 그 춘춘춘춘춘춘춘춘도와 라 데 임바간슬라 고야 완타레스 완타데 그런 거 셀레데 디제데 제튼제드레스.
So these are some of the main similarities.
데 karlana 그런 이거 바로게 탐몽도 옆에게 제주 같이레스.
Oops. That's not supposed to be it. Okay. I don't want to do that yet. Okay, let's go over the cell membrane, and then we will do an activity with the cell membrane, and then we will break for lunch.
ngasoge tapong chimu kola chinde jagin tigi jela ya tapong chimu tandebi jegu chi dubgin na tigi jela ya ne yongkon shela percent temps.
So the cell membrane surrounds all cells.
da tapong chimu tigi tapong ge ta chashiyons to pande dus chlone tamshayores.
And it's made of two layers known as what we call a phospholipid bilayer. And we'll explain that, and you will make your own here in just a second.
단다게 콜라 타풍가 치모티 그런 거 접찬이 요레스 접찬이 tilaya ngasugo uch mamage letsin yiram sa chura rambani ngune tsairete tigi jela danda chisu lagi zugu yores.
Oops, sorry. And its job is to control what goes inside and outside of the cell.
da tapunga chimu koranga liga kari cheli kari reses tambalaya tapunga na tale kari zugi chiwata tapunga na ni cholola kari alogi yobate tanzin chigi res.
The criteria for life are very specific, and if you have the wrong balance of any one thing, you can't survive.
That's 세속사 주로 세속사 되게 아니 그런 거 생리때 배팅 예전지레스로 다 대갑서 걸서고레서나 지금 나라 요비가 접사 지금 마타바도 다 뇨타바도 임바나 추니치트 그마레스.
So controlling what goes in and out of the cell is very important.
this tambalaya ani thapong nangla kari chi tama tang tang thapong nanglo ne shlo kari chi lo malo te tangjin che te ha chen ke chembures.
Okay, so we're going to just do a very brief description of this, and then you're going to make your own model of the cell membrane.
나하다 다다데라야 대신 도디 지제 자누 제라네 체주고 단다게 thapong chimuge ani basic ti che zugu res.
So a cell membrane is made up of 2 layers of fat.
다 저pongo ka chimuti bainarwa ko chi mando chi ge ramba ni ge dus tshayores.
We call this the phospholipid bilayer.
Ding mi la yo omi litsi nirum segres.
Phospho means phosphorus, lipid means fat, layer means 2 layers.
Da de kas phosphate seti da och mama setirwa. Ani lipid lighting as litsi setirwa. Tsirwa shaksirang re ani bilayers na ramba ni segres.
So the one part, this part here, this big circle, is what we call, it's the phospho part, and it dissolves really well in water.
Da de ge gang la yar mudu strip by na go ge rich towa. Rete ka resena tanda ge uch mama imbagi chashi deres da ko imba na chugna ne lamsan shudu gres.
This tail part is the lipid part, and it dissolves really well in fats. It does not dissolve well in water.
Da de ngaman jingman do chi do ate ka resena ko chugna ne yabu shugumares ina yang ko de num gna ne shasigna ne jobu shigres.
So this part likes water, this part likes fat.
Da ko go de imba na chula ga ta ko jingma de imba na ne shaksila ga gres.
Have you all ever tried to get oil to mix into water?
제주 tinda chugna la ya num tinda titi ta ya la thapsi ta nyong tinda yo chota chi nyong tinda yo es.
How long do you have to mix oil for it to go into water?
Num ti chugna ro la ya tutu kasus tang go dus.
Forever?
It doesn't mix.
It doesn't, yeah, you have to mix it forever. It doesn't work.
Nasi kanza cheba imban ko dia yo maris.
So this part is like the oil, and this part is like the water.
That is tabala degi wo ge de ko ga num tat tanda dinda do nam ton chu titi ta sa ro ati na ne degi ko jigma te ane nam tan ra ta ne gang ge chu tan ra res.
When you put these together, they orient where the sort of like this, and we'll explain this in just a second.
Da de nyam te shas tambala ya ko ga pang dik tan chi ro ta da de per ga tamba na jam pang dik res.
All of the water-loving heads, or the phospho heads, join together, and they also are attracted to the water.
Da de ka so phosphate yo sa churo och mama yo sa gi chashi de khanza ko pang dik de te ku kang gi shlo yo res na chu yo bi shlo ya te ge res.
And all of the lipid tails all also come together.
Da yang ko litsi gi topi gi juma ti samaya khusunya to pang dik res.
And because of that, you get 2 layers of these molecules with the tails together and the heads on the outside.
This tambalaya ko ge neda do ge ta tin dutte tin da ramba ni trip de te ko juma te na lo te ane go te shlo la te che ramba ni chwa chi trip ngores.
And this layer is what makes the outside of a cell.
Da da ga la ramba chi ta sha do wa de ge nga su ge thapung ge chlo chingui ji go wa thapung ge chimu te trip yo res.
Piece of paper. If I put water on this, what would happen?
It get wet.
Yeah, it would get wet. It would kind of just go into it, yeah. What if I took a crayon and then put water on the crayon?
Tanda tambu jeba na ko chute tum dugwa. Tanda ti la ya ane ko tambu te la ya choni chi dwa. Tande ti gal tele chi te ti gal la chu chi to bana kar chi gres. It doesn't dissolve. It doesn't get into the
It doesn't, yeah, it would not get into the paper. It would kind of run off. So what you and your table are going to do is you're going to take 2 pieces of paper. You're going to take a crayon. You're going to cover both sides, but completely cover them. Put them together like this.
tanda chesuge rukhana karchigure sestambalaya shogo tumbaniki shogo tumbaniki du jelaya shogo gi ngi chila roda tumbanyo rati gi ngi chite tanda delaya choni doa oti beti chonda taro chis ani tanglu jelaya ani chonda tangsu shon shanyam tendus khajaro chis.
This is going to be your cell membrane.
That to seba na ti karchigure na chonga thapong chimoti chagres.
Okay, so let's do that, and then we will see how we're doing on time. Maybe we'll finish it after lunch. Maybe we'll try to finish it before lunch. But each group, I will put crayons up here on the front and paper on the front, and I would like your table to make a cell layer.
Da tanda te choni ta shogode deja gains ne rukhare ngoni tanda ge ta thapong chimusugorwa tanda gela deshi jabati zorochis.
Okay. I believe they're in the back. Here, yeah. If you want to put those out, I can go hunt them down. Thanks. No, you can just put them together. Okay, so what we're going to do next is you're going to take your two pieces. First, make sure that they are completely covered with crayon, that they need to be completely covered.
Da chesuge shoga ni yorwa te dus kirochis da tambote kari chigore sena kogala ya choni ti cha songwa chila tangu yores yons.
We're going to put them together like this.
Da nyamte jarochis.
And you're going to get a little bit of water.
Te gala ya chutis lurochis.
I won't do it. And you're going to pour some water on the outside of your cell membrane.
Da thapong ge chimu yorwa te ge chungela ya chutis lurochis.
And you're going to see if it comes through.
Tati juchi chun dugundumundu tayares.
And it probably won't. But go ahead and do that, and then we'll talk about what that means.
Chu luchi ma dugundumundu taroches roa. Ane tege jila karimba desu jagis. Chu mabal guma da nyonya luna digirda.
If you need to get water, you can get some water from the back.
Nalo do ma those.
Is it going through the bottom? Good.
That did it.
Oh, perfect. Good.
Chelu sena chu kanza malamashi.
Very cool.
Who there?
Sorry. And each of those holes are gatekeepers, often proteins that let specific things in and out very, very carefully.
Ta tanda chechugo zu shajule chetange ta ko ge thapong chimugo nambang unila phata baimbana tanda chechu zu shajutila ye ikong phuba ro kasako ni ikong testis phuba do chita tongis tinda do chi chongo yores ta te ka res sta balaya de na ne ta de thapong chimula ta baimbana korang ta gatekeeper se do go sonyen do chi ro ta go sonyen de ge thapona ma kare tange me ya tang ma tong tila tong ji gres.
So, to be clear, water can move in and out of the cell freely.
Ta ko khasa buzu baimbana thapong ge chi chini na la ya chuti ku ta rawang gang ne jit gres.
And bigger molecules have to go through these channels that are made by proteins.
Ane teli chiwa gun du de tinda no jigu yo baimbana ane tzi ge tropi ge ta ko jilam chi gor wan de ji chin du gu res.
Okay. That's enough for now.
Okay.
That's that was a good morning. You covered a lot of territory. Let's break for lunch. Make sure to rest, and we will go over organelles this afternoon.
Da tanda shopagan zada de du stripayens tanda nyong kong persante tang te jela ne nyong kong jabla nga chulen jones tabalaya ne wang tha ko ngo to chigins.
Okay. Have a good lunch.